Hi, this is Sharon from Group 1A. Cells are the basic building blocks for all living things. The human body is composed of trillions of cells. They provide structure for the body, take in nutrients from food, convert those nutrients into energy, and carry out specialized functions. Within the cell, there are organelles. Organelles are specialized structures that perform certain tasks within the cell. A cell's command system, or nucleus, houses the genetic information, or the DNA. Long chains of DNA wrap around proteins called histones in order to form units called nucleosomes. These units condense into chromatin fiber, which condenses further to form a chromosome. Humans have 46 chromosomes, or 23 pairs of chromosomes. Cells divide in order to replenish old ones or injured ones. Most cells in the body divide continuously. The cell cycle allows the cell to grow and prepares for cell division. And when the cell divides, two identical cells are made. The cell cycle is divided into two main stages: interphase, which prepares the cell for division, and M phase, which is when the division process actually occurs. The first stage of the cell cycle is interphase. Interphase is divided into three stages: G1, S, and G2. In G1, a daughter cell enters and cellular content, except for the chromosomes, are duplicated. This is necessary to prepare the essential items for synthesis, which is the next phase. During S, or synthesis, DNA replication and chromosome duplication occurs. The duplicated chromosomes stay joined together by centromeres and are called sister chromatids. The next and final stage of interphase is G2. This is where the cell grows to have enough cytosol and cellular components in order to support two daughter cells, and the cell prepares for mitosis. At this stage, the centrioles, which organize mitotic spindle, also begin to separate and move towards the opposite poles of the cell. After G2, the cell enters M phase, which is the second stage of the cell cycle. M phase contains a few phases: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. The first phase of M phase is prophase. This is where the duplicated chromosomes condense to form a compact mitotic chromosome. This tightly associates at the centromere, which serves as the attachment site for kinetochores used in sorting chromosomes. This is also when the centrosomes begin forming the mitotic spindle and the nuclear membrane dissociates into small vesicles. The next phase is prometaphase. This is where the nuclear envelope completely fragments and the chromosomes are now in the cytoplasm. Microtubules from the centrosomes completely form the mitotic spindle and attach to the chromosomes. The chromosomes are then moved until they are all lined up in the middle of the cell, forming what is known as the metaphase plate. This phase is called metaphase. Once all the chromosomes are attached to microtubules, the cell proceeds to anaphase. The mitotic spindle shortens, splitting this chromosome into chromatids and pulling each one to opposite ends of the cell. At the end of mitosis, or telophase, two identical sets of DNA are at the poles of the cell. This DNA starts to decondense back to the thread-like chromatin structure, and a nuclear envelope reassembles around each set, forming two nuclei. After mitosis has completed, the cell then divides into two daughter cells during cytokinesis. Each daughter cell has one nucleus and contains 46 chromosomes and 46 chromatids. Following cytokinesis, the daughter cells can either go directly to G1, GS, G2, and then M phase, or the cells can enter cell arrest phase, known as G0 phase. In summary, all living organisms are products of repeated rounds of cell growth and division. During this process, known as the cell cycle, a cell duplicates its contents and then divides in two. The purpose of the cell cycle is to accurately duplicate each cell's DNA and then divide the cell and its contents evenly between the two resulting cells. As a recap, In humans, the cell cycle consists of 4 main stages. G1, during which a cell is metabolically active and continuously growing. S phase, during which DNA replication takes place. G2, during which the growth of the cell continues and the cell synthesizes various proteins in order to prepare for cell division. And the last phase, M phase, is when the duplicated chromosomes, known as the sister chromatids, separate into 2 nuclei and the cell divides into 2 daughter cells, each with a full copy of DNA.